Welcome, my dear students. So, in continuation of the recombinant DNA technology, already I hope you have watched the previous two lectures on recombinant DNA technology. Now, in this tutorial, we will talk about how a gene is cloned and what is a gene basically, and then how gene is transferred through the host cell. Okay, so this is nothing but overall the recombinant DNA technology steps, how it works actually. We'll discuss in this tutorial lecture today. So now basically what is a gene? We know in the cell, this is suppose you consider the chromosome, okay? So here are several features of a gene. The first one is a gene is a stretch of DNA that codes for a type of protein that has a function in the organism. So that means here is the chromosome. If you stretch it out, means if you zoom in, you will have such type of double helical structure which contains exon, intron, exon, and there are some promoter regulatory elements also there. This makes a complete gene. Okay. So what are the role of exons and introns? We'll discuss later on because this is just a little concept about what is a gene. So another definition of gene is it is a unit of heredity in a living organism. All living things depends on gene. That means everything is controlled by a gene. Your morphology, your physiology, your biochemical activities, your physiochemical activities, everything is regulated by genes. Okay. The genes hold the information to build and maintain an organism's cells and pass genetic trait to the offspring. That means the genes which contain a particular character that is also inherits genetically from mother to the offspring, from father to the offspring, and form a recombinant type of modes also during asexual reproduction. Okay. So next, it can be defined as the isolation and amplification of an individual gene sequence by insertion of that individual gene sequence into a bacterium where it can be replicated. So thing is that the gene is actually you cannot carry nakedly. Okay. Suppose you are having the gene in your hand, you cannot handle in the laboratory with the genes. So you need a carry, a vector. Here in biological field, we call this vector. The vector is nothing but the plasmid which carry the gene. Suppose you are going to a market, grocery market or vegetable markets, you are bringing potatoes or onion in a polythene bag or in a seeds, perforated seeds, your bag. So likewise, genes are also carries through a vector. That vector is nothing but a bacterium genome, bacterium circular genome. Okay, where the gene can be inserted. Here in the steps of gene cloning, first of all, in the recombinant DNA technology, first thing is that gene need to be cloned in a vector. So here in the step one, you can see this is the circular. First of all, this is a circular bacterial genome. Okay, this circular bacterial, a fragment of DNA in the step one containing the gene to be cloned is inserted into the circular DNA molecule called a vector to produce the chimera or recombinant DNA. This is called rDNA. So here, basically, this circular DNA is first of all cut by restriction enzyme. Then the gene of interest is inserted, means ligated by ligase enzymes and then seal it. Okay, this is the first step of cloning. Next, the vector act as a vehicle, that means act as a carrier that transports the gene into the host cell, which is usually a bacterium. Okay, here actually this is the recombinant cell. Here you can see the red portion, suppose your gene of interest you have already inserted, and rest of the things are the bacterial genome. Now this total molecule is called recombinant plasmid or recombinant genome. That genome need to be multiply in a host cell. The host popular host cell here we use E. coli is the bacterial host cell. Okay, so it is not easy to insert this recombinant molecule to insert into the E. coli host cell. There are certain process, there are certain mechanism by which we can insert the recombinant molecule into the host cell, and this process is called transformation. Via transformation, we can insert the recombinant molecule into the host cell for multiplication. In the step 3, you can see within the host cell, the vector multiplies. Here is the plasmid, because in the previous slide during step 2, already via transformation, we have inserted by heat shock or mechanical shock or some using cell lysis reagent like PEG. Okay, so we can transfer the recombinant molecule into the host cell. In the next step, we can see the host cell vector multiplies producing numerous identical copies. So clone means is the production of identical copies of the existing. So here you can see this is the E. coli bacterial DNA, and this is the circular your plasmid contains all the genes. So when the bacteria is culturing multiplied, so these plasmids are also replicating and produces more number of multiple copies. This is the step 3. Now during step 4, when host cell divides, copies of recombinant DNA molecule are passed to the progeny for further vector replication takes place. Here the simple thing, you can see the schematic diagram. This is the plasmid vector, and this is your gene of interest. You have ligated, means you have cloned. Then via transformation, you have inserted into the host cell. Here we use the E. coli is very popular host cell. This is the recombinant molecule, uh, and this is the bacterial genome, bacterial DNA. Then further amplifications of the single cell can produce multiple copies, and also inside the cells the number of recombinant plasmid also will be multiplies. So hence we can multiply the number of copies. So during step 5, after the large number of cell divisions, a colony or a colony identical host cell is produced. Each of the cell in the clone contain one or more copies of the RDNA molecule. That means after the amplification, if you culture all the cells on the LB agar plate on the selection medium, you can see there are lots of colonies. There may be a false colonies, true colonies. So you have to choose the right true colonies. That means the bacteria already contain the recombinant plasmid. So here you can do the colony PCR. This is a very vast procedure and you need not study about that. You just need the basic protocol of here. So you need to know you need to choose the true positive colonies of the plate. Then step 6, then the host cell are then lysed and RDNA can be separated. So that means it is nothing but the extraction of the plasmid extraction. So that means you are having a lot of bacterial cells. That cells already contain the recombinant molecule, and that recombinant plasmid molecule you want to extract via plasmid extraction. This is also called the host cell lysed, and RDNA can be separated. So this is the overall step. I hope you all will understand, and if you have any doubt, we will clear when we will meet. Okay, thank you for watching.